Welcome on the video visit to Luke's field experiment with Scots pine somatic embryo plants. My name is Tuija Aronen and I will tell you about this experiment and the results obtained. This experiment is part of the research that we have done to develop somatic embryogenesis as a propagation method for Scots pine. So the research from which these trees originate was done in the beginning of 2000s. So there are no breeding related aims in this experiment, but the aim was simply to study the field performance of tissue cultured Scots pine plants in comparison with seedlings. This experiment was established in 2009 on forest soil on relatively fertile site that was a former birch stand, so it's not really a typical pine site. The plants were 2 years old when planted and they were non-selected material originating in 4 open-pollinated families, or in other words, in 4 donor trees. The pine plants, tissue-cultured ones growing here, were produced by somatic embryogenesis, the explant material for embryogenic cultures being open-pollinated immature seed embryos. In this experiment, there are plants from 13 embryogenic lines, and as a control, there are also seedlings from the same open-pollinated families. And here you can see the layout of the experiment. There are altogether 10 replications that are surrounded by a buffer zone. All the replications were established with single tree plots. In the replications 1 to 5, we do have some additional material, meaning that in 6 embryogenic lines there are somatic embryo plants that originate in different types of somatic embryos. And what I mean by that is seen here. When we produced the somatic embryos for this experiment, we observed that there were different types of them. There were slim type embryos, and then there were those shorter and thicker ones that we called stub type embryos. And these embryos behaved very differently. The slim ones germinating much better and having better conversion into the somatic embryo plants. And we wanted to see if this difference would still remain under the field conditions. Therefore, for the 6 lines, we included the somatic embryo plants originating both from the slim and stub embryos and also from the intermediate type embryos. Here you can see how the experiment has grown. 1 year on the field, then 3 years on the field, and finally 8 growing seasons on the field, and the plants already start to look like the trees. But let's go now and see how they are doing today. We have measured this experiment this spring, so I can tell you fresh results. Overall viability in these experiments after 12 years at the field is 79%, so we have lost some of the plants. When comparing somatic embryo plants and seedlings, we noticed that seedlings have done a little better when it comes to viability. 83% of them is growing, while the corresponding percentage for somatic embryo plants is 78%. And there is also some variation among the plants produced from different embryo types. In a way that the biggest mortality is found with plants originating in the intermediate type embryos, only 73% of them is alive, while for the other embryo types, the viability is much closer to that of seedlings, being 80%. Now, when we walk here in the experiment, I think you notice that there are some issues in these trees. You can rather easily spot bent stem bases and also some curves in stems, and some of the trees are leaning. There is one very probable reason for that. As you remember, the plants were 2 years old when this experiment was established, so they spent maybe a bit too long time in pots. But what was worst in that was that they were grown in round pots, and this resulted in twisted roots. And after planting, these kind of roots did not provide enough support, resulting in leaning plants. Later on, the young trees or plants corrected their growth, and now we see these bent stem bases and stem curves. And of course, in some cases, also fallen trees. I would also say that these problems are worse on the other side of the experiment, where the soil is maybe more moist and soft than in the upper corner. But as you can see here, there is not much difference between the somatic embryo plants and seedlings. They both do have these same problems. You can see some differences between the seedling families and also some differences among the SE genotypes, but overall all of them, they do have a certain percentage of trees showing either the bent stem base or the curves in the stem. Then, what about the growth of the somatic embryo plants and seedlings? Are there any differences? It is of course difficult to see it in these single tree plots, but let's look at the measurements done. So, first we have here old data on the first 6 years on the field. I would actually like to draw your attention to the fact that when we planted the material on the field, the seedlings were significantly bigger than the somatic embryo plants. And this difference remained during the first 6 years at the field. But now, when we look at the fresh measurements for the both height and stem diameter, what we can see is that although there is some variation, all the statistically significant differences between the somatic embryo plants and seedlings have disappeared. So in other words, in all the families, the seedlings and somatic embryo plants have grown equally well. And the same is also true for the trees originating in the different somatic embryo types. As you remember, there were some differences in the viability of these, but when it comes to the either height or stem diameter after 12 years on the field conditions, no differences related to the original embryo type. So, to summarize. In this field experiment, we have seen that the somatic embryos of Scots pine, that you can actually see here on the plates, and that were produced by tissue culture in laboratory, after they have been germinated to form somatic embryo plants, or emplings as you can also call them, as seen here being transplanted into bigger containers, can after acclimatization be successfully planted and grown under field conditions. And they really are able to grow into trees. And their growth is equal to that of seedlings, despite the fact that these somatic embryo plants were smaller than the seedlings at the time of planting. And obviously, as already told, we can also see in this experiment that in order to avoid curling of the roots and subsequent leaning of the plants, resulting in bent stem bases and curved stems, one should never grow the pine plants in round pots. But then, on the other hand, based also on the present results, it seems to be not very critical how the original somatic embryos look like if they only are able to germinate, since we could not see significant differences in the growth of the plants produced from the different embryo types. If you wish to get more information on this field experiment or the tissue culture process used to produce the somatic embryo plants, you can look at these references. Thank you very much for your attention.